In the last 5 decades, we've learned a lot about the secret lives of proteins, how they work, what they interact with, the machinery that makes them function, and the pace of discovery is accelerating. Just this week, the company DeepMind shocked the protein structure world with its accurate, AI-driven predictions. But the 3D structure is often not enough to truly understand what a protein is up to, explains Ken Dill, director of the Laufer Center for Physical and Quantitative Biology at Stony Brook University and a member of the National Academy of Sciences. It's like somebody asking how an automobile works, and a mechanic opening the hood of a car and saying, "See, Terry's the engine, that's how it works." In the intervening decades, computer simulations have built upon and added to the understanding of protein behavior by setting these 3D molecular MAC. Analyzing their energy landscapes, interactions, and dynamics has taught us even more about these prime movers of life. We're really trying to ask the question, how does it work? To be useful, simulations need to be accurate, and to be accurate, simulation needs to progress atom by atom and femtosecond, 10 to the power of -12 seconds, by femtosecond. To match the timescales that matter, simulations must extend over microseconds or milliseconds, that is, millions of time steps. Since 2015, with support from the National Institutes of Health and the National Science Foundation, Dill and his team have been working to speed up molecular dynamics simulations. Their method, called MELD, accelerates the process by providing vague but important information about the system being studied. Dill likens the method to a treasure hunt. Instead of asking someone to find a treasure that could be anywhere, they provide a map with clues, saying, "It's either near Chicago or I." This narrowing of the search field can speed up simulations significantly, sometimes more than 1,000 times faster, enabling novel studies and providing new insights. Protein structure predictions for COVID-19. One of the most important uses of biophysical modeling in our daily lives is drug discovery and development. 3D models of viruses or bacteria help identify weak spots in their defenses, and molecular dynamics simulations determine what small molecules may bind to those attackers and gum up their works without having to test every possibility in the lab. That's the primary thing that we used Frontera for. The Frontera supercomputer at the Texas Advanced Computing Center, TACC, the fastest at any university in the world, allowed Dill and his team to make structure predictions for 19 additional proteins. Each of these could serve as an avenue for new drug developments. They have made their structure predictions publicly available and are working with teams to experimentally test their accuracy. While it seems like the vaccine race is already close to declaring a winner, the first round of vaccines, drugs, and treatments are only the starting point for a recovery. As with HIV, it is likely that the first drugs developed will not work on all people, or will be surpassed by more effective ones with fewer side effects in the future. Dill and his Laufer Center team are playing the long game, hoping to find targets and mechanisms that are more promising than those already being developed. Now there is a push to transfer this concept to target SARS-CoV-2 proteins. Collaborating with Stony Brook chemist Peter Tong, they are working to simulate the interaction of novel proteins with the COVID-19 virus. For one simulation we need 30 GPUs and 4 to 5 days of continuous calculations. The team is developing and testing their protocols on a non-COVID test system to benchmark them. Once they settle on a protocol, they will apply this design procedure to COVID systems. Every protein has a story to tell, and Dill, Brini and their collaborators are building and applying the tools that help elucidate these stories. Please support our channel to grow by pressing subscribe button as well as the bell icon for daily science updates. Thank you.